Okay, we are moving on to our next unit, which is involving DNA and protein synthesis. So what we're going to cover at the beginning of this is what is DNA and the structure of it. We talked about in the chemistry of life unit that DNA was a nucleic acid, so that was a polymer of things called nucleotides. All right, and if you don't remember, DNA stands for deoxyribonucleic acid. Okay, so this is going to become important a little bit later because the deoxy and the ribo is going to show up in its structure. Okay, so what is DNA? So it is actually a polymer of the monomer for nucleic acids, which we said were nucleotides. All right, so if you look over here, this is a strand of DNA on this side over on the right. This is it all wound up. If you've seen the one on my desk or the one that I painted earlier this year, you'll see the kind of twisted helical structure here. And it's made of two strands that are twisted up exactly like this. If you look over on this side, this is an untwisted version, and it kind of shows you what's on the inside and what's on the outside parts. So if you recall from the chemistry of life unit, when we talked about a nucleotide, we said that there was a couple different parts to it. So the specific parts for these are the phosphate, the base, and the deoxyribose sugar. All right, so in a nucleotide, one nucleotide contains the phosphate, the sugar, and a base. All right, these things will always stay the same. The phosphate and the deoxyribose sugar will stay the same for any of our parts of our DNA. This part over here, the base, is what changes. Okay, for DNA, our sugar is always going to be deoxyribose. So that's part of the reason why it's called deoxyribonucleic acid, because it's a nucleic acid, which is the polymer of deoxyribose sugars. Okay, so we're gonna, when we get to a little bit later, towards the end of this unit, we're gonna talk about something called RNA, which is ribonucleic acid, which has just a ribose instead of a deoxyribose. Okay, so this is what makes DNA, the deoxyribose sugar right here. Okay, so what about those bases? So I said that those things can change. The phosphates and the sugars will not, but the bases change. So if you remember, we said DNA is the way that we encode all the genetic information for your body. So this is going to tell your cells how to work, what pigments to produce, what hormones to release, enzymes, all that stuff is included through the arrangements of just these four things. All right, so your entire body functions correctly due to the way this is stored with just using 4 units. Okay, so that's totally cool. So what are these 4 bases? So these are called adenine, thymine, guanine, and cytosine. Okay, just those 4. If you look over here, so if you notice any structure differences, so this here, these are the bases on the inside. So we have guanine and adenine. If you notice, these both have 2 rings included in them. We're going to call these 2 purines. Okay, so Purines. All right, the one way to remember this, so adenine and guanine are an A and a G. The chemical symbol for silver is Ag. So pure silver purine, or like angel has an A and G in it. Angels are pure, so if you want to remember it that way. Okay, so purines have 2 rings in them. These bases are also sometimes called nitrogenous bases. And the reason is, if you look in here, in all these bases, there is a nitrogen, at least one. There are actually multiple. There's at least 2 in actually all of them. Okay, so that's why they're called nitrogenous bases. They have nitrogen within the base. The other 2, so if you look over here, we have cytosine and thymine. Those only have 1 ring. So we actually call these pyrimidines. Okay, so that is going to be T and C. Okay, the way, instead of having to write out adenine, thymine, guanine, cytosine every time, we just use the first letter. So A, T, G, and C. Okay, so how are all of these organized? So we've already gone through nucleotide. All right, so the nucleotide had the phosphate, the sugar, and the nitrogenous base. Now, remember, nucleotides are the monomers for nucleic acids, so they are the building blocks of DNA. So what's going to happen is these nucleotides, as units, are going to connect together and build up DNA. So if you look at this picture over here, we have our bases on the inside, and we have our phosphates and sugars on the outside. Now, the way that these get arranged in a DNA molecule is your phosphates and your sugars end up being in this like backbone outside area of the DNA helix. So this is normally called the sugar phosphate backbone. Okay, so these things are always going to be on the outside. On the other hand, we have our nitrogenous bases then that line up on the inside. Okay, and remember, these are the combinations that help store that genetic information in the codes for how things function. If you notice in both these pictures, the pictures before and this one over here, there's a pattern to the way that these bases line up. If you notice, there's always an adenine and a thymine joined together. Same thing over here. And then the cytosine and guanines always pair together. And they're over there. Okay, so this is always, 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 always the case. So A pairs with T, and C pairs with G. All right. The one cool thing, if you notice, we talked about hydrogen bonds in the chemistry of life unit. The differences between these pairs that are bonded together is with G and C, there are 3 hydrogen bonds. One right there, and one right there. So this would be 3 bonds. With A and T, there's only 2 bonds. All right? So that's kind of how they go together. And if you remember, adenine and guanine were our purines, and cytosine and thymine were our pyrimidines. And so we always have a purine pairing with a pyrimidine. It's always the case. There is no way that you're going to have an A ever pair up with a G or a C ever pair up with a T. Okay, they have to go like this, otherwise your DNA molecule won't bond together correctly. Okay, so this whole thing, how in the world did people figure this out and when? This didn't get discovered, the structure of DNA, until 1950s, so that wasn't that long ago. Our major scientists that helped discover this, so we have on our left Rosalind Franklin. And what she was doing is she was doing something called X-ray crystallography. And what this does is it takes pictures of molecules at the very, very tiny level. And she was working on this. This is called Photo 51. This is like one of the most important photos in science. And while she was working on this, there were two guys over at another university, James Watson and Francis Crick. James Watson is actually still alive. And they were trying to figure out the structure of DNA. And unfortunately, they were trying very, very wrong, and they were trying to make 3 strands instead of the 2 that we know is there now. Rosalind Franklin was the one who enabled them to finally figure out the correct structure for DNA. This photo, once you did some math and some very detailed calculations, showed that this was a double helix structure and that there were only 2 strands in this DNA molecule. So this allowed Watson and Crick to figure out the reason why their model wasn't working, that their base pairs actually had to be on the inside, and there was only 2 strands. So without Rosalind Franklin, it might have taken those 2 a lot longer. However, these 2 got the Nobel Prize. She did not. Unfortunately, she died before the Nobel Prize was awarded for this discovery, so she could not get awarded one posthumously, so that means after she died. So only these 2 guys got credited with it. And one cool little last thing, I like this quote from her: "Science and everyday life cannot be and should not be separated." Right? You should be looking at science in your everyday life and making sure that you are evaluating things like a scientist. Okay? So that is our overview of the structure of DNA. We're going to then go on to replication, translation, and transcription later on in this unit, and then finally, mutations.